Okay, Assalamualaikum and a very good afternoon. So at least now is 6 p.m. here. So today we're going to a lecture on glycolysis. I'm sure if you are from biology background during your matrix, you should be already familiar with this term. Otherwise, if you are not from biology background, we can always go through this more personal. All right, we can discuss about this later on. Okay, so glycolysis is actually the breakdown of glucose. Glyco is actually glucose and lysis is breakdown. All right, so for the learning outcomes, I hope you can read it yourself. It's pretty straightforward. Okay, so for the first slide, I just want to show you the metabolism maps of macronutrients. As you can see from here, all the macronutrients, which are the first one carbohydrate, the most common one, second is protein, and the third one is fat, they are all eventually will merge and be converted into energy in TCA cycle and also electron transport chain. Alright, and this is the example of the glycolysis reaction and also its reverse reaction, which is gluconeogenesis. So I think the gluconeogenesis is the one in green, and gluconeogenesis, which is the formation of glucose, which only happens in liver, is actually in orange in color. Okay, so glycolysis can happen in all the cells of the body, but gluconeogenesis, which is the formation of glucose, only happens in the liver and a small amount also in kidney. And you can also see here there is a special color which is green, and others are black and green as well, because this try to show you that these three reactions are the most important reaction in glycolysis, because this point of reaction is actually can be regulated. Means if suddenly your body wants to speed up the glycolysis, they can speed up through these three green reaction. Or if they want to slow down, because probably our body already got a lot of ATP, a lot of energy, so we don't have to break the glucose anymore, they can slow down through this reaction. And the same thing for the reverse reaction as well. Okay, you can see the one orange in color is actually the one that can be regulated in gluconeogenesis. And if you notice, you can also see actually the reverse reaction, the regulated points, is actually the one that is reverse in glycolysis. For example, the one that is regulated is reaction number 1 in glycolysis, but the one that is regulated in gluconeogenesis is the last reaction, which is the mirror of the glycolysis. Okay, you can see here as well, it's actually the mirror of this one. And this one you can see 2, but this is only 1, because in gluconeogenesis, in order to bypass this reaction, you need to undergo 2 reaction instead of 1. Okay, what I'm trying to say is, from let's say from reaction 1 to 2 requires 1 enzyme, but to reverse it, it needs 2 enzymes to form the same product. Okay, we will go through about this one in more detail in gluconeogenesis. Okay, so this is the site of reaction. Like I said, this is the shorter version of the notes, and I emphasize this site of reactions because it is very important for you to know where the glycolysis and gluconeogenesis happens. Okay, if you can see from here, the glycolysis happens in the cytoplasm, means that it doesn't have to have oxygen. But if you want to go through to the next part of the reaction, which is Krebs cycle or TCA cycle, and also electron transport chain, you must have oxygen. Otherwise, the reaction will stop at pyruvate, and this pyruvate will be converted to lactic acid. And this lactic acid will be transported to the liver and converted back to glucose. Okay, so this is anaerobic reaction. The glycolysis, but Krebs cycle and etc., they are both aerobic reaction, happens in the mitochondria, because this is, as you can see, the organelle that contains double membrane. So the only organelle that fits the description is mitochondria. Alright, so the trivia is mitochondria exist in all cells? Okay, the answer is no. There is a cell, a type of cell that doesn't have mitochondria, which is red blood cell, because we know red blood cell transport the oxygen, right? And inside the red blood cell there is no organelle at all. So they don't have mitochondria. Means that they only perform glycolysis to get their energy. So red blood cell will always produce lactic acid. So that's why our body always produces lactic acid. But if we exercise, we will produce more lactic acid because we don't have enough oxygen. So in the muscle cell or in the cell that is exercising, we only perform the glycolysis. If not, how the cells going to generate energy? Okay, the answer which is through glycolysis. But the energy that is formed during the anaerobic reaction is much less than aerobic respiration. Okay, only 2 instead of about 30. Okay, ATP is being produced inside mitochondria. How to transport it out? Okay, you can find this in electron transport chain lecture later. Where did the mitochondria comes from? Okay, this is just a side note. Like I said, this is trivia, so this is like a fun fact. So mitochondria is actually thought to be a type of bacteria that fuse into our cell in a process called as endosymbiotic. Okay, so this bacteria is the bacteria that can produce the energy. So when they fuse with our cell change them into through evolution, and then the cell will change them into an organelle that can produce the energy. So there are many evidences. You can read it yourself in Google or Wikipedia. Okay, so this is the important part of glycolysis. I will also put a video from YouTube here so you understand about it more. Okay, so as you can see from here, glucose is 6 carbon molecule, and then it will produce the first product from the transferase or group transfer reaction to produce glucose 6-phosphate, and eventually it will be break into 3 carbon molecule. So 3 carbon molecules times 2, we get 6 lah. Alright, so we get 6 again. So that's why after this reaction, you need to times 2 for everything that you get. Okay, for example, I ask you guys to calculate the number of ATP in TCA cycle. You need to times by 2 if I mention glucose, because you need to break them into 2 here. Okay, so there are 2 parallel reactions that are happening at the same time. Alright, so the first one that is important for you to know is you need to know the type of the reaction types. You don't have to remember everything, because I know in matrix probably you need to draw and remember everything, but from here you just need to know, if I show you guys glucose to glucose 6-phosphate which involve ATP, what type of reaction type is this? Okay, so you can take a look at one is the coenzyme one, and the second one for the first one is the involvement of coenzyme or any other molecule such as ATP, and the second one is the involvement of the enzyme. So every time you see the word kinase, means this is going to be a transferase reaction, and if you see the kinase, usually it will be involve ATP, because one of the phosphate from adenosine triphosphate will be transferred to this molecule. So from glucose becomes glucose 6-phosphate from 1 phosphate from ATP. So because we take 1 phosphate from ATP, it becomes adenosine diphosphate, 2 ATP instead of 3. You can see the name of this enzyme as well. Isomerase means this is an isomerase reaction. You can see the name kinase, and you can see the molecule ATP. So this is going to be a transferase reaction. You can see aldolase. Okay, this one probably a little bit hard to know what type of reaction, but if you see the molecular, you actually know this is actually a lyase reaction. This is isomerase. Every time you see the word dehydrogenase and the coenzyme such as NAD+ or FAD, this is usually what we call as redox reaction. Okay, I think you will know about this in my previous lecture. Okay, when we learn about the 6 type of reaction. I'm not a chemist, so I don't really know as well. So I just remember the trivial part of the reaction. So you need to know probably more about the pathway rather than the specific type of chemistry that happening inside the glycolysis and all other reaction as well, right? So what are the most important reactions and why? I already answered before. The most important reaction is actually reaction number 1, reaction number 3, and reaction number 10, because this is the regulated point. But the most important reaction is actually reaction number 3, because we call this reaction number 3 as committed steps. It means that once the glucose enter into reaction 3, it is committed to produce pyruvate. If glucose enters and come up to glucose 6-phosphate, it is not committed to produce pyruvate yet, because there is other reaction that can happening here and produce, for example, they can produce glycogen, they can produce other stuff as well, amino acid and so on. But once once it pass reaction number 3, it must produce pyruvate. That's why we call this one committed. I usually give the example of marriage. Once you are engaged to that person, you are not committed to live your life together with him or her yet. Means that probably you just passed the reaction number 1, the first regulated reaction. But if you are married already, you are actually past reaction number 3, you are actually committed to finish your life together with him or her. And also, the important thing to know that each of these regulated points, 1, 3, and 10, they have their own characteristic, right? The first characteristic is it must be irreversible. Oh, sorry, I think that is the second characteristic. The first one is it must have high negative energy. I think you learned together with Professor Nazami, like, about the endogenic reaction. Okay, so the negative delta G. So here 1, 3, and 10, they all have high negative delta G reaction. The second characteristic is they must be irreversible. It is a given, because if you have a nice and a big negative energy, usually it is very spontaneous, for example, from glucose to glucose 6-phosphate, so it cannot reverse. Okay, so it is irreversible. And the characteristic number 3 is, remember, there are 3 characteristics for the regulated points, the most important reaction in glycolysis. The third characteristic is actually if this product tries to produce back its substrate, they must use a different pathway or a different enzyme. For example, glucose 6-phosphate, if it wants to produce glucose again, it needs to use a different enzyme compared to hexokinase, which is in this case is glucose 6-phosphatase. Okay, and one of the example that you can see from here is actually reaction number 10. Phosphoinopyruvate to pyruvate just require one reaction, but from pyruvate to phosphoinopyruvate in gluconeogenesis, actually you need 2 reactions. From pyruvate, it must produce oxaloacetate first, then oxaloacetate will produce phosphoinopyruvate. That's why in this reaction, you actually can see there, for gluconeogenesis, there are 1 extra step. Instead of 10 reaction in glycolysis, you need actually 11 steps in gluconeogenesis. That's why you will have 2 regulated points in gluconeogenesis, because there are 2 reactions. Okay, 2 reactions from pyruvate to phosphoinopyruvate, and both of them in gluconeogenesis is actually regulated points. Okay, so the third thing that is important about glycolysis is energy investment. So the first 5 is energy investment, and the last 5, they are called as energy payoff or energy harvesting stage, because for the first 2 you need to use ATP, 1 ATP, 2 ATP, right? But in the last 5 you will produce back 4 ATP, so you need to minus 2 from 4 ATP you produce during glycolysis. You need to minus by 2 to get net 2 ATP. The same as you invest your money in the stock market. You invest 1,000 and then you get 2,000. So means that you get a profit of 1,000. So the final product will be 2 ATP and also 2 NADH. You need to remember, because they are actually break here. Actually, they only produce 1 ATP and 1 NADH here. But because there are 2 molecules now, you need to times 2. So you need to get 2 NADH and 4 ATP. Sorry, 1 NADH and 2 ATP here. So you times by 2, you get 4 ATP and also 2 NADH. And you need to minus by 2. Okay, I stop here. I will continue in the next. I will show you the YouTube video, and then we will continue with the next lecture. Alright, the same lecture. Okay, thank you.